My name's Tim Miller. I work at the Institute of Plant and Food Research in New Zealand. I'm currently a doctoral student at the University of Otago. The work I'm going to be talking about today has also been partly funded by the Tools for Polyploids project, so a big thank you to all the fiscal sponsors of the project. Hopefully we've got Susan Thompson in the chat who will try and answer any questions while we're going. If there's a question that she can't answer, just keep in mind it's my fault for not writing enough documentation. Okay, so I'll start with a little bit of motivation of why we're looking at haplotype assembly from sequence data. Then I'll go over some of the theory. There's a little bit of statistical theory to go over there. Finally, I'll try and do a quick live demonstration of the tool on a small data set and then talk a little bit about some future research. So if I could sum up the motivation for this tool in a sentence, it would be to try and make most of the data we already have. So we have quite a lot of historical sequencing data and as well as an increasing volume of new sequencing data at Plant and Food. Historically, we've used a lot of SSRs. I'm not really looking into their use at all. They've actually been very successful in kiwi fruit, which is a highly polyploid organism. However, we're looking to more modern sequencing NGS techniques. So we have got whole genome sequencing for quite a few of our more important specimens, but obviously this is very expensive and not applicable for when sequencing hundreds or even thousands of samples. So more recently, we've been looking at reduced representation sequencing, mostly working with rapid genomics. So we have quite a lot of capture sequencing, which is what my PhD work has all been based on. However, more recently, we've been looking at FlexSeq, which is slightly sparser again than CaptureSeq, although the sort of early data we're getting from that seems to be very promising. It looks like it's higher quality than the CaptureSeq even. So these are the sort of areas we're looking in future for more sequencing data. I'll just start with very quickly summarizing a couple of the problems we have working in kiwi fruit. I'm not going to give a big background into kiwi fruit. I actually think Gibran gave a really nice overview yesterday, so thank you for that, Gibran. We're primarily working in diploid, tetraploid, and hexaploid populations. These, as Gibran pointed out yesterday, roughly correspond to the hexaploids being the traditional green kiwi fruit. The tetraploids, most of the tetraploids we're working with are the gold kiwi fruit, and some of the newer red golds that you're seeing on the market are diploid. So we're working with a lot of ploidy ranges. There are some complex interactions between the ploidy levels as well. It is possible to produce interploidy crosses. Some of those are naturally occurring. The other thing we have with kiwi fruit is they're very variable, so very high level of variation across the genome. This isn't surprising considering how polyploid they are. Also, the genus as a whole is not that well understood taxonomically. There's multiple species complexes, and most of the commercial fruit that you'll eat come from the Chinensis species complex, which is previously described as multiple different species, but now more recent treatments have treated them as subspecies that can intercross, and there's different ploidy levels in the different subspecies, so it's a very complex group. These graphs are just showing sort of the number of variants we get within these bait regions that we're working with, and it is variable in the different cohorts, of course. Here you can see that we actually get the highest variation in the tetraploids, or at least with these cohorts that were examined here. But commonly within a fairly small bait region of 120 base pairs or so, we commonly get 10 to 20 SNP variants. So it's a very variable organism. This is a plot showing some of the capture data for a single organism at a single bait target. And so you can see this really sort of typical, almost bell curve shape to the read depth that we get with captured GBS. This is because you have a capture bait targeting a specific locus on the genome, and we get really deep sequencing right over that locus, but you also get a sort of a trail off of overlapping reads out to the sides as well. These brightly colored dots on here are SNP variants with respect to the reference genome. So each gray bar indicates a read, and the brightly colored dots indicate a variant or a variation with respect to the reference genome. If I zoom in on a section of this, you can see these very clear repeating patterns in here. So we see this really obvious one of red, yellow, blue. We also see other ones like this just single red variant on these reads. These are almost certainly the underlying haplotypes within the organism that you're seeing here. So the reads are generated from, the read fragments are generated from the chromosomal copies in the organism, and for any given chromosome, this is a tetraploid, so you'll have 4 copies of that chromosome with different patterns of variance along each copy. So the aim of this tool is really to try and assemble the reads into something that resembles those underlying chromosomal copies, at least within short segments along the genome, rather than just calling the SNP variants themselves, because if we just call the SNP variants, we lose this linkage information between the different pairs of SNPs. To explain that in a bit more detail, I've put together an example just with a trio here, and what we're really interested in a lot of genetic work is IBD, so identity by descent, where 2 organisms share the same allele because they have their allele has come from a shared ancestor. So what you can see with a variant like this is actually just the SNP here alone could have indicated, at least in this case, that this allele is IBD with this one here. But you can imagine if we start to have a larger pedigree with more founders, a single SNP might be able to identify you and link you back to a single ancestor. However, when you that will only tell you something about that single ancestor potentially. So by taking these informative SNPs and merging multiple informative SNPs into a single haplotype marker, we start to actually get a marker that can help us differentiate more and more between multiple ancestors. The other thing you'll notice here is that with a single biallelic SNP, there's potentially only 2 sort of allelic phenotypes you can have. You can either have a homozygous, or you can have homozygous for the reference or for the alternate allele, and then you can have heterozygous. When you have a heterozygous call in a tetraploid or haploid organism, when you're dealing with a biallelic marker, it's very uncertain what the dosage is. So those 2 alleles could be in a dosage of 1:3, 2:2, or 3:1. By merging multiple SNPs together, we can actually start to pull out more information about the underlying dosage. And you can see that really clearly in this parent here, where we have a whole bunch of biallelic SNPs pushed together, but because each of them tells you something different about the organism, we can actually see that this individual has 4 distinct alleles, and so we know it's got a dosage of 1:1:1. Now, this isn't perfect. We are working in restricted regions, so you will see in these other 2 examples, we have a dosage of 1:1:2, but even a dosage of 1:1:2 tells us a little bit more than if we were just working with a biallelic SNP alone. So we developed, as part of my PhD project, a tool for micro-haplotype assembly. When I say micro-haplotype, I'm using that term rather loosely. This tool can assemble longer haplotypes, so we're mostly working in about 120 base pairs. How long you want to assemble, how sort of the size of the region that you want to assemble will really depend on the level of variation you have. So with the data I'm working with, I could actually expand that region to be larger, but given the high density of variance, I would prefer to keep it at about 120 base pairs and focus on trying to improve the quality of this assembly. And the reason for this is this tool is really about generating markers for statistical genetics. So there are quite a lot of haplotype assemblers available that work in polyploids. A lot of these are very good tools. I think where ours differs mostly from many of the others is that we're focused on assembling haplotypes in repeatable regions from the same basis variance, so we can have comparable alleles across multiple different analyses of multiple different data sets. We also wanted a tool that's suitable for a range of sequencing platforms. So in the first couple of slides, I showed the different sequencing platforms we use. We would like a tool that can work across Capture-GBS, across FlexSeq, and across whole genome sequencing, and potentially even some other sequencing platforms as well. And that's really about making the most of the data we already have. So even if we move to a really good sequencing platform in future, something like FlexSeq, we'd still like to be able to use the data we already have. Finally, it needs to be a tool that's suitable for complex populations under selection. And when I say populations here, I'm really talking about very intricate and reticulate pedigrees. So the under selection thing is particularly difficult because many of the other phasing tools that are available, so particularly Some of the tools that we talked about yesterday that are designed for working in mapping crosses, they're relying on assumptions of Hardy-Weinberg equilibrium, or they're essentially relying on the fact that this is an unselected cross to help improve the phasing of that data. When you're working with highly selected populations, particularly when they're small populations, you might only have a handful of examples of a cross between two parents. You can't really rely on that as much. So currently our tool is just doing essentially sequencing of individuals, although it can do it to multiple individuals at the same time. But each individual is assembled by itself without respect to the others. That does bring in some problems of its own, which I'll get into later, but it does mean that it's flexible for use in a whole variety of situations. So the tool itself is a Bayesian assembler, so it's based on Bayes' theorem, which I've shown here. The important things to take away from this equation is what we want to find out is this thing on the left, which is the probability of a genotype given in a set of observed reads. And what Bayes' theorem tells us is that this is proportional to these things, the numerator on the right, which is the first symbol here is the likelihood function, which is pretty much the inverse of the posterior probability function over here. So the likelihood function is saying, what is the probability of generating these reads from a proposed genotype? And the second symbol up here is the prior distribution. So this is what is the probability of a genotype without any reference to the reads at all. And that's where we can start to include some additional information that we have a priori about our genotypes. So for the prior distribution, we're actually using the Dirichlet multinomial. If that doesn't mean something to you, don't worry about it. The important thing about the Dirichlet multinomial is that it does let us include some prior information, particularly around the expected inbreeding of the sample that we're assembling. So a more inbred sample is more likely to have homozygous genotype. And so particularly when we're working these highly selected advanced individuals, it's really important that we have included that inbreeding coefficient in there so that we can help that to improve the quality of the assembly. Using this prior also theoretically lets us include some information that we know about the expected allele frequencies. So if we have some prior information about the allele frequencies for that population, we could in theory include that as well. That's currently not implemented in the tool, although it is something we are planning on looking at in future. The main reason that that's not implemented yet is when you're assembling haplotypes from scratch, you often don't really know anything about the prior distribution of haplotypes. So until we have a good use case for it, it's not really a priority to implement. The second important term on that numerator was the likelihood function. So this is a big equation. It's actually fairly straightforward once you break it down, but the main takeaway from this equation here is this 1 over k here, which is where k is the ploidy of the organism. And what this is telling you is that the tool we've built is assuming that the reads are generated roughly in proportion to the underlying haplotypes. Now that's an assumption, it's an important one. It's an assumption that's also not entirely true all the time. Different sequencing platforms do have bias towards some alleles. Without including additional information from the pedigree and so on, we can't really do anything other than make this assumption. So this is sort of the best case. What this means is if you do have bias in your underlying sequencing towards a specific allele, is that our tool may actually, it will probably still produce the right haplotypes, but it's quite likely to produce them in the wrong dosage unless it's a fully heterozygous or fully homozygous genotype. This is one of the areas of the tool that we need to benchmark a bit more to gain an understanding of, but it's also a difficult area to benchmark because it is specific to the sequencing platform and the organism that you're working with. The tool is using a Markov chain Monte Carlo simulation to do the actual genotype calling, hence the MC and MCHap. This is a simulation algorithm where we take in a bunch of observed reads. Then we start proposing genotypes here. And so we run this simulation over many iterations, and each iteration a new genotype is proposed based on the previous genotype, and that new genotype is either accepted or rejected based on the likelihood function and the posterior distribution, the prior distribution. Now, over many runs of the simulation, over many steps in the chain, the frequency of genotypes that we see in this trace on this side starts to approach that of the posterior distribution. What that means is when we are calling haplotypes and assembling haplotypes with MCHap is that we are not just calling the best haplotype, we're actually learning something about the probabilities around that. We're not just calling the best genotype, we're recording information about the probability of that genotype. And MCHap is capable of outputting the entire posterior distribution over all of the genotypes it's looked at. That can get messy, so I'll talk a little bit more about that later. The tool itself has been made publicly available as of earlier this week, so you can find it on our GitHub page following this link up the top. I don't think that's in the description for this talk, so I'll make sure it gets added in the next few days. The tool is called MCHap. There's a little bit of information from the README there. It's been released under an MIT license, so it's very available for commercial use or anything like that. There are a couple of asterisks attached to its release. The first is that it is still in a relatively early version, so it's at version 0.5.1, which is a beta release. That means that we are pretty happy with the feature set that's available, but there still might be some minor changes, particularly around default parameters and that sort of stuff. And we are hoping to get some feedback to help us make those sort of final adjustments. The second thing is that the tool, we haven't actually published in the academic paper yet around this tool. So the tool is available, but it might have some rough edges, and there's not currently a citation that you can cite for it. Also, it hasn't undergone any sort of peer review yet. That being said, if someone is interested in using it, would really love to get some feedback. You can ask questions or give me some feedback either via emailing me directly or leaving an issue on the GitHub page. Issues on the GitHub page are great because then other people can see them as well and learn from your questions. The asterisk attached to that is I am currently in the last couple of months of my PhD, so I might be slow in replying to you, but I do promise I will get back to you eventually. Next, we're going to quickly flick to a live example. Hopefully this works. We're actually going to look at two tools. So MCHap itself is really a collection of small tools. The main two tools in it currently are Assemble, which is used to do the de novo assembly, and that will produce a VCF. But we can also run MCHap call, which is a very similar tool, except it's used to call genotypes from a set of known haplotypes. So MCHap assemble is used for de novo assembly from basis SNPs. MCHap call can be used to call haplotypes from a set of reference haplotypes. So I'll just switch to my terminal. Can you see this?
Yes.
Yes,
cool.
we can.
Yeah, we see it.
Okay, so MCHap is it's quite similar to a variant caller in some ways. So it's not so much an interactive tool like we saw yesterday with many of the tools that were presented. This is more a tool that you set up with some parameters at the command line and you let run, and then you do your own filtering and your own analysis of the results. So in this directory, you can see that I have a couple of bash scripts, assemble.sh and recall.sh. You can also see there's some directories that hold some input files, and there's a list of BAM files. So if I look at the list of BAM files, you can see that this is just a plain text list of BAM files that are in that BAM folder. The reason I have this list is I find it easy. Yeah?
Sorry, Tim, before you go on, I think Peter was suggesting if you could zoom your screen a little bit. Maybe the text is a bit small for some people.
I might have to restart my terminal to do that.
Just control and scroll up or down.
Oh, okay. I did not know this feature. Thank you. Okay, so we have this directory of, is that clear enough for people?
Yeah, it's better.
Okay, thank you. So I'm going to start by looking at the contents of this first script. Okay. So what we can see here is that we're running MCHap assemble, so we're using the assemble subtool, and then we have this list of parameters that we're passing to it. So I'm actually going to start the script running before I talk through these parameters, because it does take about 40 seconds to run. Okay, so the the input files are all listed at the top here. So MCHap takes a target, a bed file containing genomic targets. So this is just a standard bed file containing genomic intervals. In this case, I only have 10 intervals in here because it's a small example. This can be a 3-column or a 4-column BAM. If you use a 4-column, bed file, if you use the 4th column of the bed file, which contains a name for each loci, then you'll see that name printed in the output VCF as well, so that you can link your variants back to the intervals that you've assembled. The second input here is actually a VCF file. So MCHap doesn't identify SNPs from scratch. It uses an input VCF file that contains SNPs, and it will assemble haplotypes from those SNPs. That does mean there's an additional step in there where you need to do your own SNP calling. In this case, I've used Freebase. The advantage of that is that one, you can use a good established tool like Freebase to do your SNP calling. It also means that you can use the same set of reference SNPs over multiple different analyses, and so you get really repeatable results and comparable results between analyses. An important thing to note about the SNPs VCF is that it doesn't actually need to have any sample data in it. We're only looking at the reference and alternative alleles. So that means that it's actually quite easy if you want to generate your own SNPs VCF from scratch, or else you can just use the output from something like Freebase. The next input here is a reference genome. This isn't actually used in the simulation itself. This is just used to identify which allele is the reference allele, and it's also used to fill in the blanks between the SNP variants they're assembling, so that when we open the output VCF, you'll see that we have these long haplotype alleles that don't just contain the SNPs we assembled, but also contain the reference genome between those SNPs. The next input is the BAM list that I've used. So here I've just used a plain text file with a list of BAMs because it's simpler to show in a small example like this. If you want to, you can also just use --BAM and import a list directly into MCHap like that. So you just have a long list of BAM files here. The next argument is the ploidy. So in this case, I'm actually assembling a small tetraploid data set. This is because it's a nice, convenient, small data set for working with. It's also a published data set published by some of our colleagues here at Plant and Food. The ploidy can be specified per sample if you want to. So MCHap can deal with any ploidy up to about 20x. That's not actually a theoretical limitation, that's just a practical limitation to make it easier to work with internally. If there is a demand for ploidies higher than 20, I'd be interested to hear. I actually think at that point, the tool would probably start struggling to assemble things anyway. But it can deal with any even-numbered or odd-numbered ploidy up to 20 at the moment. The next argument is the expected inbreeding coefficient of our samples. In this case, I've just taken a bit of a guesstimate for this example. Again, you can specify an inbreeding coefficient per sample. So if you're dealing with something like a pedigree that you're trying to assemble, you can actually calculate the inbreeding coefficients and then specify a single coefficient per sample. The next two arguments are around the expected error rate in the sequencing data you're working with. So by default, MCHap will just use the Phred scores associated with each read in your BAM file. That's something that I might change in future because the Phred scores used on their own will actually significantly underestimate the sequencing error in a lot of cases. So if you do have an estimate for of the sequencing error for your reads that you're working with, it's much better to specify that precisely and then use ignore base Phred scores to ignore the base Phreds. Incidentally, doing this will also give you a bit of a speed up in your algorithm, because when you have just a constant base error rate among all your reads, there's some things MCHap can do to make itself faster. The next two arguments are arguments to do with the actual MCMC simulation itself. So as I described earlier, a Markov chain Monte Carlo simulation has runs for a number of iterations. So we're specifying the total number of iterations we're running for here. And then we're removing the first 500 of those iterations as burn-in. Essentially, this is because usually when you have a Markov chain, it doesn't start in an ideal location and parameter space. So you want a while for it to sort of warm up and get into a good area of parameter space. And so throwing away the first segment of the chain is usually a way of improving the accuracy of your posterior probability estimates. Finally, this argument here is quite an important one. So I know it's a bit of a mouthful, haplotype posterior threshold. Essentially, if you set that value to 0, then MCHap would report every single haplotype it looked at in all of those samples that it's assembling. The problem with that is there can be hundreds of thousands of haplotypes, and if you include all of those in your VCF, you have a very messy and hard to interpret output. So this parameter is essentially a threshold, a posterior probability for individual haplotypes that they need to exceed. And so when we run through those simulations, we calculate the posterior probability of each individual haplotype, or the posterior allele frequency you can think of for each sample. And we require that a haplotype allele exceeds that threshold for it to be included in the output VCF. Finally, we have this cause argument. In this case, I'm just running on a single core. MCHap is very capable of running on many, many cores. So the maximum number of cores that it can run on is defined by the number of targets in your bed file. In some cases, I've done assemblies where I have 800 tetraploid samples over 10,000 loci. In those cases, I think the highest number of cores I've used is around 600 or 800 on a compute cluster. In a case like that, you probably wouldn't use the cause argument. So this is good for when you're working on a single machine, like on a laptop or a desktop. When you're working on a cluster, there are other ways you can make this tool run in parallel that are more efficient, because it can be spread across multiple different machines. I haven't got that example of that here today, but there is an example of how to do that in the repository on GitHub. Finally, we're doing some stuff with the output of MCHap here. So MCHap will output a plain text VCF file. So what we're doing here is we're piping that into BGZip to compress it, and then we're running tabix to generate an index for that VCF file. This isn't necessary. You can just save the plain text VCF if you want, but I think this is good practice to automatically compress and index your VCF file. So we'll just open the file, the results from that assembly. And have a look at the VCF file. So I've tried quite hard to be very compliant with the VCF standard, and I think it's a reasonably good result. So we have some metadata in here about the VCF version, the date the file was created, the version of MCHap that was used, and we even saved the command that you used in MCHap. So this is all really good for sort of reminding yourself what you did and how you did it later on. Next, we see a bunch of contigs. So these are our chromosomes that are in our reference genome. In this case, we're just assembling a few loci on chromosome 5. Next, there's a bunch of info fields. So a lot of this, if you're familiar with VCF output, a lot of this will look familiar. The main one I want to draw your attention to here is snivpos, which is a variable length field, and it tells you the positions of your basis SNPs within each haplotype that you've assembled. So if you want, you can use that to break your haplotypes back into the SNP calls. If I jump down here, I think we've got just under 40 samples in here, 38 samples. And so a single record in a VCF file from MCHap looks something like this. So that's one line that's just been wrapped around because of the space in the terminal here. Now, so this is the chromosome for this variant, the base position for the start of this variant, and this is the name of the locus that we assembled. So this is taken from the bed file and returned in the VCF file, so you can identify your individual loci. So this sequence here is the reference allele. So we're assembling 120 base pair regions. which means we have these really long variants. It's not particularly nice to look at them in a VCF file like this. But then following that, we have our alternate alleles. So in this case, we have 4 alternate alleles that are also 120 base pairs each. And then we have the allele counts for the alternative alleles. So we can see we have 55, 45, and 15. Down here, we have the genotypes themselves. I won't stay on here too long because I'm just looking at the time and I don't have that much longer left. But if you're familiar with the VCF file, this will look very familiar, other than the variants are probably larger than you're used to seeing. So the next tool we're going to look at is MCHap call. So this is the next script, and I'll just try and get that out from behind the red mark. So MCHap call requires an input VCF as well, except in this case, instead of using a VCF of SNPs, we're using a VCF with known haplotypes. And so we don't have a bed file here of loci because it just uses the intervals already defined by the haplotypes in this VCF. Most of the rest of the arguments are the same as the assemble tool, except there are fewer arguments that are required here. The additional argument of putting in here in this case is the genotype posteriors flag. So this will produce the full genotype posteriors based on the input haplotypes. So start that one running. This tool typically runs a little bit quicker than the assembly tool itself, because that's essentially because there's a smaller parameter space for it to examine. So one of the advantages of the recall tool is that it's faster. The other advantage is that it can produce more accurate results. So if you have a if you have a set of reference haplotypes, then you're reducing the parameter space that MCHap has to look at, which means you'll get higher posterior probabilities for MCHap recall than you will for the assembly tool. And a lot of the time what we do is we will actually run a de novo assembly on a bunch of related samples, and then we'll immediately run them through MCHap recall or MCHap call. The reason for that is the assembly is done on every individual sample, and for some of those samples, the assembly might not work that efficiently, and so you'll get some bad variants in there. Because we're working in a group of closely related things, we expect them to share a lot of alleles between one another. And so when we rerun and recall them using MCHap call, we're looking at the haplotypes that were assembled across all samples and using that set of haplotypes to recall all of the samples. So if you have a sample that had a bad assembly in the first case, it's very likely to get corrected by the haplotypes that were assembled in its related individuals. zcar isn't a command. So this VCF looks very similar to the previous one. The big difference you'll notice when we get down here is the output is much longer. So we have these final argument, which is the final record for each assembly is this long list of zeros with a single one in it. So this is the posterior distribution. In this case, because we have, thank you whoever got rid of that, because we have very good read depth and relatively few SNP variants in this case, we're having a very high probability for our posterior mode. It won't actually be exactly 1, but we're rounding to about 3 decimal places here just to try and keep the output size relatively small. And so we have a posterior probability or an approximation of an posterior probability of 1 for the assembly for this individual. If I scroll down here, we will start to see some that have a bit more variation than that. Although it might be hard to find because this is a relatively low variance data set. So in this case here, I'll just point out, we only have a reference allele and no alternate alleles. So everything has been called as homozygous for the reference. That's because for this loci, when we first did the assembly, there were no SNP variants within that region. And you'll see that a bunch of the other outputs that are sort of quality control metrics are blank here because they can't be calculated unless there's some alternate alleles. So MCHap will always return a genotype for each sample. That can be a genotype with a very low posterior probability. So you are required to do some checking of your own results here, and the posterior probabilities are reported in the output as well. I won't worry about this posterior mode right now. Oh, here we go. So what we can see here for the posterior distribution of genotypes of some of these individuals is that we have a long list of low probabilities. So this is, I won't say a bad assembly, but this is probably an example where we have relatively low read depth, so we have a very uncertain result. And so by the actual genotype call itself for this example is 1233. I wouldn't have a high faith that that's a correct genotype there, but it's the best guess given the data that we have. So in this case, it would be worth looking at the posterior distribution to get an idea of the sort of range of possibilities. That being said, if you have a much more variable data set than this, that list of posterior probabilities for every possible genotype can get enormous. And so I would highly recommend that you assemble your data set without the genotype, the full posterior distribution first, and then look at the number of variants that you have in that data set and think about the number of potential genotypes there are, which can, as I say, be enormous. Okay, we've only got a few minutes left here, so I'll quickly switch back to my PowerPoint. Just a quick thank you to Sam Baldwin.
You have another 10 minutes.
Yeah, thank you. Just a quick thanks to Sam, Katrina, and Susan, because they were the ones that initially generated and published that data set. And it has been used in a couple of different publications, including some from the Wageningen group, I believe. So these were my backup slides in case that live demonstration went badly. So I'll talk now a little bit about some of the metadata that we output, some of the more important output fields in that VCF. So we're using a Markov chain Monte Carlo simulation, as I said. Typically when you run a Markov chain Monte Carlo simulation, you'll look at some quality metrics for the trace. You'll look to get an idea of whether your Markov chain's converged or not. And when I say converged, we run these simulations in duplicate, and you can actually run more than 2 simulations each if you want. And then we compare the results of those replicate Markov chains to see if they've found the same result, so to see if they agree with one another. So one of the parameters that we output in MCHap is this MCI tag, which stands for Markov chain incongruence. In this case, a value of 0 indicates that there was no evidence of incongruence, and a value of 1 or 2 indicates incongruence. There's a little bit of a difference there, but either way, it indicates incongruence. So what incongruence means in this case is that we have run the same analysis twice, and not only have we found different results, but there was reasonably strong posterior probability for both of those results. So by default, that threshold is 60, a probability of 0.6, I should say. You can adjust that threshold as well as you want, but that seems like a sensible starting point. And what we can see here, so this is the assembly example I just showed was running over 10 loci. This is running over all around 1,500 loci for this chromosome. And we can see here the proportion of incongruent assemblies we got across the chromosome. So you can actually see there's a large number where we have sort of more than 10% of the assemblies were incongruent. If you look at the total number of assemblies by the total number of samples and look at the number that were incongruent, it's only about 1% of the total assemblies that are incongruent. But when we look at the mean incongruence across each loci, like we're doing here, what you can see is that for some loci, there's a lot more incongruence in the assemblies than there are for other loci. There's a couple of reasons that can be. One is that we didn't run the MCMC for long enough, and that it will improve the assembly if we rerun it for a longer period of time, so a longer number of iterations. Another example cause could be that you have some sort of contamination in your data, or that you have some sort of copy number variation that you're unaware of, and both of those will cause issues because you've told MCHap the number of haplotypes you expect to find when you specified the ploidy, but the true number of haplotypes that appears to be there is actually more than the ploidy. So this can also be a real issue if you specify the wrong ploidy for an organism. So if you try and assemble a hexaploid as a tetraploid, it will give you a tetraploid genotype as a result, but you'll almost certainly find that there's been some Markov chain incongruence. Now, these results on this first panel are for the assembly tool. When we reran that tool, reran those assemblies with the recall tool, we see a lot less Markov chain incongruence. And this is again because the parameter space is smaller when you're using the recalling tool, which means that you generally get a higher quality result because there's less possible genotypes for it to examine. You still see here that 3 of these loci we're getting between 10 and 20% of the assemblies are looking like they're incongruent. So I would be highly suspicious of those loci in that case and do some more investigation as to whether there's some evidence of copy number variation or whether they're just very variable and difficult to assemble loci, and I would consider removing those from any downstream analysis. The second parameter we're going to look at is MEC, or minimum error correction. So this is quite a common metric used in a lot of haplotype assembly tools. We don't actually use MEC in the assembly itself, but we do use it, report it just as a QC tool. So the minimum error correction here is calculated as the number of corrections that would need to be made to your reads to get them to match your genotype perfectly. And the minimum error correction will almost always be above 0 because there's inherently a low level of inaccuracy in Illumina reads. Illumina reads are very accurate, but there's always a small number of missed incorrect base calls in there. And what we've plotted here is the proportion of, so minimum error correction divided by the total number of base calls in that data set. So we get it out as a percent that's comparable across multiple loci. You can see that this loci that we had a lot of Markov chain incongruence, we're also getting quite a lot of, quite a high MEC score for as well. Again, a bit less obviously now, but we do see a slight improvement in performance with the recalling tool than we do with the assembly tool. It's a smaller difference here, probably because there's a certain amount of error correction that's always going to be inherently there because of the errors in the Illumina reads. We also have this K-McCov, so K-McCoverage metric. This is very similar to minimum error correction. It differs a little bit in that instead of looking at the full length of the haplotypes, we're now looking at, in this case, 3 MERs across the haplotype. So 3 MERs comprising 3 different SNP variants within the haplotype. And this can give a bit more of a fine-grained look if that's necessary, although it typically reports a very similar result to the minimum error correction. It's kind of the inverse here because this is specified as the proportion of K-MERs among your reads that match the one or more of the haplotypes in your genotype. And so in this case, a value of 1 indicates that all of the K-MERs match your genotype. So just a quick summary of the features that are available so far. And as I say, the documentation is a bit minimal at the moment, so we are hoping to improve that. But we have 2 basic tools in there to assemble haplotypes from scratch or to recall or genotypes from a set of known haplotypes. MCHap can handle any ploidy up to 20x at the moment. That can theoretically be extended even higher. I'm not going to do that unless there's a real demand for it because it gets a bit more difficult to work with. And I do think that even if you're assembling a 20x organism, that you would have such a sort of diffuse posterior probability distribution that I'm not sure that this would be the best tool for you anyway. We can use an inbreeding prior, and I think this is really important, particularly when you're working with those more inbred advanced crosses. If you don't include that inbreeding prior, what's likely to happen is that your genotypes will get called more heterozygous than they really are. Because there's a certain number of errors in your read data, what can happen is, particularly when you have lower coverage, if you have a couple of error reads in there that look similar to one another, then that can look like an additional haplotype. So by including the prior information about an inbreeding coefficient, you're more likely to get a accurate assembly. We do have a mechanism available in there called parallel tempering. This is something that can be done with Markov chain Monte Carlo simulation. Essentially what this is a way of improving the mixing and the posterior distribution at quite an increase in computational expense, so it requires running a bunch of Markov chains in parallel. I wouldn't recommend doing this for every sample in a large data set, but it can be specified on a per sample basis. So particularly if you have some really important founders where you want to have high confidence in the assemblies for those founders or other important individuals, then you can specify parallel tempering for those individuals. We can output full posterior distributions, but as I say, the VCF that's produced can be huge, so be careful with that feature. We have relatively good performance. So MCMC is an inherently computationally expensive method. I think we've achieved reasonably good performance here. So the tool is written in Python. However, we're using a special JIT compiler for the actual MCMC process itself, which means we get much better performance than you would with standard Python. The number JIT compiler is using very similar technology to the Julia JIT compiler, if people are familiar with that language. More importantly for performance is that we can run this in highly parallel situations. And so this tool has really been designed to run in, efficiently in an HPC environment. And finally, and I think possibly one of the most important features, that we've made a real effort to use standard bioinformatic formats here. So we're really using VCF, BAM, BED, and FASTA. There are a few plain text additional inputs that can be used, like a list of BAM files. All of those additional plain text inputs are very simple tabular plain text, so they're quite easy to create and interpret. Some future work we're wanting to do, there needs to be a lot more documentation done. So as I say, this was only released earlier this week. Once I finish my PhD, I plan to write a lot more of the documentation for this tool. There also needs to be some more simulation analysis done there as well. We would like to include the option of specifying a prior for allele frequencies. As I say, this isn't a theoretical limitation currently. We know how to do it. It's really just the difficulty here is how to input that prior for allele frequencies in a sensible way, because we don't want to create additional input files if we can avoid it. Yesterday, Jeff talked about posterior allele frequencies or posterior dosages as an input to GWAS poly in future. Currently, we can't output something like that, but that is planned as well. So we would like to be able to, rather than put out a posterior over genotypes, put out a posterior distribution over alleles. And that should be much smaller than the full posterior and much easier to work with in downstream applications. So we're looking at a couple of options for outputting that. Finally, and this is a much more stretchy goal, and it's not guaranteed to happen, but in future, I would like to look at the option of including some pedigree data in there. So currently, as I said, we're assembling each sample individual. If we did have some pedigree information in there, that could help improve the dosage calls in some cases. So I just wanted to finish with this slide here, which is showing the assembly results for a large complex pedigree I've been working in. Each of these nodes represents a genotype, and you can see down the bottom, or in some of these founders as well, we have allele calls for a single locus. And if you look closely at this, what you'll see is some of the allele calls for some of these advanced samples don't make sense. This sample here has allele number 8 in it. If you look at the founder material for that sample, none of those founders have allele 8 in it. So unless there's been some mutation, which is quite unlikely, that's almost certainly a miscalled genotype there. If we included some prior information and some pedigree information, we could potentially avoid those sorts of errors and improve the dosage, as I said. So that would be something I'd like to do, although there's a lot more theoretical complexity to adding that feature, and so it may or may not happen in future. Finally, I just want to thank particularly the SCRI tools for polyploids. This is a great event, and also the fiscal sponsoring for some of my time working on this assembler. The other important people to thank is the Kiwifruit Royalty Investment Program, which has paid for a large part of my PhD. My supervisors and advisors, the Kiwifruit breeders, the actual real breeders that produce the organisms I work on, and the market development team who produce the data that I work on. So a big thank you to all of those and the rest who have helped me along the way. Are there any questions?